Presentation on DNA versus RNA. We're going to be looking at their function and structure as well as the nucleic acids. So what is the difference between DNA and RNA? Well, RNA is a single-stranded structure usually, and its main goal is to transcribe and translate genetic information usually found outside of the nucleus. There are many different types of RNA. Messenger RNA, or mRNA, makes an identical copy of the DNA, replacing thymine with uracil, and delivers this message to the ribosome inside of the cytoplasm. rRNA, or ribosomal RNA, will read the message inside the ribosome and prepare sections of mRNA codon for the transfer RNA. Transfer RNA, or tRNA, will deliver the matching amino acid based off of its complementary anticodon. DNA stores genetic info. It's usually in a double helix structure, and it's found inside of the nucleus except for mitochondrial DNA. Its goal is to transmit hereditary information to offspring, and its difference between DNA and RNA structurally is that there is an H atom present at position 2 instead of a hydroxyl. So the missing oxygen here allows it to form a double helix structure, and you will not find uracil inside of DNA. As a protection mechanism, when uracil shows up in a DNA strand, uracil DNA glycosylases, or UDGs, will recognize this base as mutated and remove it from the strand. So it's important to remember when matching the strands, the 3 to 5' leading strand will always run opposite to the 5 to 3' lagging strand. As you can see here and here, here and here. And then here's the difference between the riboses. Nucleic acids, well, they are the monomers of RNA and DNA. So they're composed of nucleotides, which are all composed of one nitrogenous base, either adenine, cytosine, guanine, thymine, or uracil, a phosphate group, and a pentose sugar. So these are going to be covalently attached to one another. And then in DNA, you'll see that the nucleobases will actually have a hydrogen bond to one another. So you can see here a tetranucleotide, which means there's 4 nucleotides stacked together. These can create a chain of polynucleotides, which will eventually become a nucleic acid, which will form our DNA and RNA. There are a couple different types of bonds within DNA and RNA molecules. We have the phosphodiester bonds, which are the backbone of the molecule, and this will be covalently bonded to each other, and it'll be 10 times stronger than the hydrogen bonds that the nucleobases have. So strong covalent bonds will protect the genetic material inside of the DNA and create a negative electromagnetic charge. The phosphate uses 2 of its arms to link the 3' in the sugar molecule to the 5' location on the next nucleotide. Hydrogen bonds, which are found between the nitrogenous bases, there's usually 2 or 3 of them depending on the base pair, are about 10 times weaker than covalent bonds. So it's not a true chemical bond. The weaker bonds allow the helicase to unwind the strand and expose the nitrogenous bases of the area that's being copied. So electrostatic forces between the 2 polar molecules, either adenine-thymine and guanine to cytosine or adenine to uracil, will hold the bases together. So now that we've covered the double and single helix structure, ribose versus deoxyribose, and the phosphate groups, we can take a look at the nucleotides, the nucleobases. There are 5 main nucleobases within our DNA and RNA structures. The purines, adenine and guanine, have much stronger intermolecular bonds than pyrimidines due to their two-ring-membered structure. They have a melting point of 214 degrees, making them less susceptible to UV damage. And then we have the pyrimidines, which bond to the purines with hydrogen bonds. So those will be cytosine, thymine, and uracil. They have a single ring structure, making them much more susceptible to UV damage, having a melting point of only 20 to 22 degrees Celsius. So the first base pairing we're going to look at is adenine and thymine, which is a base pairing that's found only in DNA. So adenine, or adenosine monophosphate, is found in both RNA and DNA, but binds with thymine only in DNA. AMP can phosphorylate into ADP and ATP, which is essential for energy production and life. Adenine is also known as vitamin B4, which has been classified as non-essential and is no longer an actual vitamin, since we can synthesize it. It's found in meat, fish, and eggs. Thymine, also known as thymidine monophosphate or thymidinic acid, is very sensitive to UV radiation. It can create dimers when adjacent thymine nucleobases are covalently bonded to each other. UV sterilization works by creating these dimers to inhibit DNA replication in bacteria. Unrepaired dimers can lead to melanoma. The base pairs are pretty close in their molecular structures, so any small alterations can lead to mismatched base pairs and mutations. For example, the demethylation of thymine at the 5' creates uracil. That's the pairing of guanine and cytosine, which occurs both in DNA and in RNA. So guanine, or guanosine monophosphate, or guanilic acid, can undergo deamination and become xanthine, the precursor to 7-methylxanthine, the active metabolite in caffeine. Caffeine works by blocking adenosine receptors. Usually adenosine binds to these receptors and slows our neural activity. But caffeine has the same purine shape as adenosine, and it can bind easily to these receptors, blocking the process. Cytosine, or cytidine monophosphate, is found in RNA and DNA. It can deaminate to become uracil, which will increase the likelihood of C to T mutations. And then you can see here that cytosine plus a methyl group is equal to 5-methylcytosine. 5-methylcytosine plus H2O becomes uracil. So it'll just be a hydrolysis reaction. So finally, we'll be looking at the base pairing of the purine adenine with the pyrimidine uracil. This pair only occurs in RNA. So in RNA, uracil actually replaces thymine, and methylation of uracil at the 5' location can actually create thymine. So this is just looking at how similar these nucleobases are, and just by aminating, deaminating, methylating, demethylating, we can have a completely different nucleobase. And this is just to show how simply mutations can occur. These are my sources that I used, and thanks for listening to my presentation.